to Dr. Nur Sharmila and my fellow classmates. My name is Dinisha and my matrix number is 223193. So today, me and my groupmates are going to be discussing about the impact of genetic modifications and stress conditions on protein expression. So we will be discussing about the first factor, which is genetic modification. There will be three categories, which are plasmid A, plasmid B, or control. For second factor, it is going to be about stress condition, where there will be two categories. First one is heat shock at 42 degrees Celsius, and second one is oxidative stress. So oxidative stress is basically an imbalance of free radicals and antioxidants that leads to cell damage. So now, my groupmates are going to discuss further about this topic.
Good day to Dr. Sharmila and to all my classmates. My name is Emil Asni, matrix 222785. I am from Bachelor of Sciences in Cell and Molecular Biology with Honours Year 2, Semester 1. Today, I'll be presenting about the experimental unit and the variables. The experimental unit, which is a cell, which is the smallest independent entity that a treatment is applied to. On other cases, a culture or an organism can be used too. On the other hand, protein is not used as an experimental unit, but it is used to measure an outcome. This is because treatment is only applied to a living system and protein are not independent entities. The proteins are the production occurs and the cells are the functional unit of life, where the gene expression for protein is stored. Now, let's talk about variables. Independent variables, which are the factors manipulated in the experiment, that includes genetic modifications, which refers to specific alterations in the genetic makeup of an organism. Not only that, stress conditions, which refers to environmental or physiological stresses such as temperature, pH, and other variables. The dependent variables, which is the measured outcome, would be protein expression levels. This involves both quantitative and qualitative measurements of proteins produced under varying conditions. Techniques such as bloating, ELISA, and others can be employed for this analysis. That will be all for me. Thank you for your time, and I'll pass it to the next presenter.
What is completely randomized design and why we use it? CRD is used to randomly assign the experimental units, which is cell, to the treatment groups, combination of genetic modification and stress condition, to see the effect on protein expression. The total of treatment combination is 4, which is genetic modification A plus heat shock, genetic modification A plus oxidative stress, genetic modification B plus heat shock, and genetic modification B plus oxidative stress. 3 replicates are made to get reliable results. Hence, the total of experimental units is 4 treatments multiplied by 3 replicates equals to 12 experimental units. So how do we get the numbers on the table? After exposing the experimental units to the treatments, protein expression are measured. The number are made by using Excel random function to ensure that the treatment are done randomly.
Hello. So today I'm going to explain the concept of a two-level factorial design. So two-level factorial design is a powerful tool for analyzing how different factors influence the outcomes of the data. So it's particularly useful when you want to study not only the individual effects of the factors, but also how they interact with each other. So first of all, what is a two-level factorial design? So a two-level factorial design is an experimental setup where we investigate multiple factors, each at exactly 2 levels. So for example, let's say we are studying about the effect of genetic modification and stress conditions on protein expression. So our 2 factors would be either genetic modification or stress conditions. So next is why do we use this two factorial design? So this design is particularly efficient because it only requires fewer experiments compared to testing all possible combinations. So other than that is that it allows us to analyze both main effects and how factor influences each other. So how does the two-level factorial design works? So we choose the factors that we want to study, and we define their two level of factors. So for this sample, we use genetic modification. Is either wild type or modified, and the other one is like for stress, is either non-stress or with the gene is added with stress. So the second one is we run the experiments for all possible combinations of these factors with two factors at two levels. So next is that for each of the combination we measure the outcome, and in this case we want to look at the protein expression. So after we do the experiment, we analyze the results. So once the data is collected, we calculate the main effects to see how each factor affects the outcome of the experiment. So for example, does genetic modification increase or decrease the protein expression? And then next is that the stress condition affects the expression level of the protein. So we also analyze the interaction effect to understand if the effect of one factor depends on the level of the other factor. So for example, does stress have a stronger impact on protein in genetically modified organism compared to wild type? So here we have the example result. So let's say we measure the protein expression levels under the four combinations. The first one is wild type, no stress, the result is 50 units. The second one is wild type with stress, 70 units. Next is modified with no stress, 90 units. And the last one is modified with stress, 120 units. So from this, we can calculate that the main effect of genetic modification, how much protein expression changes between wild type and also modified. So the main effect of stress is that the change due to stress versus no stress. So why is this important? Is because this design is widely used in science and also engineering, because it helps us to uncover hidden relationships between variables. For example, for this experiment is the genetic modification and also the stress condition. It is efficient, easy to implement, and it gives us deeper insights into complex systems. So for example, in biology, it can help identify conditions that maximizes protein expression, while in manufacturing, for example, it can optimize the product quality. So, other than that, in conclusion, a two-level factorial design is a simple but also powerful experimental framework. So by testing factors at two levels, it helps us to understand not only the individual effects, but also the interactions between factors. Whether we are in like biology, engineering, or any other field, this method can save time and also provide valuable insights. That's all from me.
Next, I will talk about comparison between characterized random walk design versus two-level factorial designs. It has six aspects. For the first one, the purpose for CRD, it used to compare the effects of different treatments without considering interactions between factors. For true LFD, it focused on examining how multiple factors and their combination affect the outcome, including interaction. For the second aspect, the number of factors and levels. For CRD, it can handle any number of factors and levels, but through LPD, it only work with multiple factors each at exactly two levels, example height or low. The third aspect is factor interactions. For CRD does not account for interaction between factors. Uh, for 2LFD is specifically designed to study both main effects and interactions. The fourth is experimental runs. For CRD, it requires fewer runs, especially with fewer factors. For 2LFD, it requires more runs as the number of factors increase due to testing on factor combination. The fifth is flexibility. For CRD, highly flexible in terms of factors and levels. For 2LFD, less flexible as it is limited to 2 levels per factor. Lastly is analysis. For CRD, it's analysis often using ANOVA to compare treatments means. For 2LFD, uh, it's more complex analysis using pattern ANOVA to examine main and interaction effects.
So these are the advantages of both designs. For CRD and also 2LFD. CRD stand for completely randomized design and 2LFD stand for two-level factorial design. For the first aspect, which is simplicity, for CRD, it is more easier to design and implement, while 2LFD, it most likely more complex but structured for the multifactorial design or studies. For the second aspect, which is flexibility, for CRD, it more accommodates any number or treatments without restrictions, whilst 2LFD, it more efficiently handle multiple factors and their combinations in a systematic way. For the third aspect is interaction analysis. For CRD, it is not designed to detect or analyze interactions, while 2LFD, it is explicitly identifies and quantifies interaction effects between the genetic modification and also stress factors. And the last aspect, which is the fourth aspect, is efficiency. For CRD, it requires many runs to study multiple factors or combinations because it cannot runs for the many factors, so it need many runs to study for the multiple factors. While 2LFD simultaneously examines multiple factors with fewer experiments, saving time and also resources. But why 2LFD is more suitable for this topic? It is because protein expression under the genetic modification and stress involve multiple interacting factors which 2LFD can effectively capture. As you know, 2LFD can use for the manufacturing studies. And the second reason is 2LFD can quantify main effects and interactions, which is it will revealing how genetic and environmental factors combine to influence outcomes. And the last reasons is the efficiency of 2LFD can reduce the experimental burden while maintaining the robust results.
So right now I will explain about these advantages of completely randomization design, CRD. So first, it less accurate result because unrestricted randomization. Because of the greater influence of the random choice, so the randomization may introduce imbalance. Second one, it only be applicable in a small number of treatments. This is because it will be hard to control the variability. Next, it will be not suitable for heterozygous material because this can lead to bias. Because the treatment being unevenly distributed across the unit, which verify the characteristic. So it leads to the biased result. Next, the sensitivity of the experiment will increase as the number of error of degree of freedom increase. Okay, so right now I will explain about the disadvantages of the two-level factorial design. So first is it's difficult to handling the noise and the confounding, which means because if interaction between factor and external variable are not controlled, it will easily become misleading. Next is its lack of curvature direction, because curvature direction will increase as increased level being tested. So since this is only 2 level are tested, so it's low curvature direction. Lastly, the 2-level factorial design, it is limited to linear relationship because the design only capture the interaction between 2 points. So if the relationship between 2 factors are not linear, so it may result in the incomplete conclusion. Completely randomized design (CRD) are suitable to use when the experimental setup is simple, interactions are not important, and the experimental units are homogeneous, while the 2-level factorial design are suitable to use when multiple factors and their interaction need to be studied, and even it requires more experimental runs. So in conclusion, if the system is complex and the aim is to optimize and understand the interaction between the factor, the 2-level factorial design are generally better compared to the completely randomized design.